Good afternoon, ladies and gentlemen. My name is Ryan Sutar. I'm one of the senior investment managers here at Sharewise, and we're here with Dr. Kilian Murphy, the managing director and CEO of Cynata Therapeutics. Thank you very much for your time, Kilian. Really appreciate the opportunity here. Now, today's just going to run through fairly simple. We're just going to have a quick presentation from Kilian, and then we're going to open the floor up to a Q&A. So I've got a couple of questions here that we'll get us started with, but if any participants throughout the presentation would like to submit questions through the Q&A function on Zoom, feel free to do so, and I'll ask them to Kilian after the presentation. Kilian, it's all yours.
Right, thanks, Ryan, and thanks for the opportunity to talk here today, and thanks to everybody for joining. So, as Ryan said, I'm going to run through a presentation. I'll try not to make it too lengthy, but of course, this presentation is available on our website, and you can read it at your leisure, and feel free to get in touch after the fact if you have any other questions as well. So Cynata is a clinical stage biotech company based in Melbourne. We are exclusively focused on our stem cell manufacturing platform, which we call Cymerus. And this is really a revolutionary way to make stem cell therapies at scale. And just to take a step back, some of you may be familiar with mesenchymal stem cells, or MSCs. These are cells that have been used therapeutically to treat a wide range of illnesses, especially over the last 15 years or so. They've been used in a lot of clinical trials and shown really a lot of promise. The problem has been conventional manufacturing methods rely on an ongoing supply of new donors, and that's associated with challenges with consistency, potency, and scale. And I'll explain that a bit more in a moment, but that's really the problem that we're trying to solve. And our patented Cymerus platform uses another type of stem cell, and sorry for all of the terminology here, but it's induced pluripotent stem cells, or iPSCs. And by using iPSCs, we can overcome that major obstacle. And this allows us to produce an effectively limitless quantity of consistent and high quality MSC doses from a single blood donation. Again, I'll explain that more in a moment. We've generated compelling clinical data using these cells already in graft versus host disease and diabetic foot ulcer. And again, I'll explain more about that shortly. And we've now got a rich clinical pipeline. So we've got 3 randomized controlled clinical trial readouts coming up between late 2024 and early 2026. So we've got lots of clinical data to come. And we're also about to start another clinical trial imminently in patients who are undergoing a kidney transplant. So we've had a lot of progress this financial year, which of course is coming to an end just now. We've completed enrollment in 2 of our randomized controlled trials. That's in osteoarthritis and diabetic foot ulcer. We've announced further encouraging clinical efficacy data. This is the preliminary initial data from our DFU trial, which we released in February. We've started our phase 2 graft versus host disease trial, enrolled the first patient in March this year. And as I said, we're about to start the kidney transplant trial. And we've also strengthened the management team. So we've created a new position of Chief Business Officer, which has been created really with an eye on the next stage of the company's growth, specifically with regard to business development and partnering. And we've recruited Dr. Matthias Kroll, who joined us in April this year. He's got a wealth of experience in this area, and I'm very confident he's going to be a great addition to the team. So moving forward with the platform and why it is we're doing this. So as I said, MSCs can be used for a wide range of illnesses, and they have been over the last 15 years or so in particular. The reason they're of interest mostly is they have an immunomodulatory effect, which means is they balance the immune system. And that's important because immune system reactions, especially excessive immune reactions, play a role in a lot of different medical conditions. And these cells also promote tissue repair and regeneration, which has benefits, of course, in a wide range of illnesses as well, notably diabetic foot ulcers, which is one of our ongoing clinical programs. But again, the challenge to date has been how you manufacture these cells consistently and at scale. And so this brings us to the iPSC part of the story. Now, iPSCs, or induced pluripotent stem cells, are adult cells that are reprogrammed to behave like pluripotent stem cells. So they behave like embryonic stem cells, but they're not derived from embryos. They're derived from an adult donation, in our case, a blood donation. And what pluripotent means is that the cells have an effectively limitless proliferation capacity. So they can just keep producing more and more of themselves. So you can produce an effectively limitless number of cells from one donation. And these cells also have the potential to turn into any other cell type. And in our case, that means MSCs, but they can also be used to generate other types of cells. Now, importantly, because they're derived from adult donors, we avoid all of the ethical controversies that you may have heard of related to embryonic stem cells. So none of that is relevant to us. So we believe that iPSCs are an ideal starting material for the commercial production of cellular products. Now, this slide just explains how our platform differs from a conventional approach. So at the top, this is a conventional manufacturing process. It involves getting a tissue donation, for example, bone marrow or fat, and then isolating the MSCs from that donated tissue. And they then have to grow those cells in the lab to generate enough cells to do whatever they want to do. And that's a process called culture expansion. But now the challenges with this are, first of all, it's logistically challenging because you have to get an ongoing supply of new donors. And donating bone marrow, for example, is not a straightforward thing to do. It's much more invasive than, for example, donating blood. But perhaps more importantly, it's well established that there's lots of variability in MSCs derived from different people. That's probably not surprising, but it's a very big problem because with a pharmaceutical-like product, you need to be able to show consistency from batch to batch and over time. The other issue that people have is in order to generate enough or as many cells as possible from a single donation, there's a tendency for them to put the cells through a very extensive level of culture expansion. But that's a problem because it results in a loss of functionality and loss of potency. Now, our process overcomes those challenges because by using iPSCs, as I mentioned, we can generate essentially all of the cells we ever need from a single blood donation. So we don't need to get new donors. And that means we don't have that variability. Also, because we get our scale at the iPSC stage of the process, we don't have to put the MSCs through a great deal of culture expansion once we form MSCs. So we avoid that loss of potency as well. And when you put all this together, it means we have a way of making as many cells as we want from a single cell bank with a high level of potency and consistency. And I should just quickly mention that we have patent protection protecting this process as well. Now, just to quickly mention, we have a strategic partnership with the Fujifilm Group. Fujifilm is a very big player in the manufacture of products like this. We are currently in the middle of getting our manufacturing process set up at one of their facilities, which is called Fujifilm Cellular Dynamics. And Fujifilm is also a shareholder in Cynata. So this partnership really is important for us as it provides us a means to move towards a genuine commercial scale manufacturing. Now, moving to our clinical data. So our first clinical trial was conducted in patients with graft versus host disease, which I'll explain in a moment. But it's worth noting that this trial has led to 2 publications in Nature Medicine. Nature Medicine is one of the most prestigious and high impact journals worldwide. So this really is quite an achievement for a phase 1 trial. And I think the key reason that it got this level of prominence is that this was the first clinical trial worldwide with an iPSC-derived product. And as I mentioned earlier, iPSCs can be used to produce all types of therapeutic cells. So we're not the only ones pursuing iPSCs. In fact, there's lots of companies now who've come to the same realization as us that this is the way to make cells at scale. But we were the first to complete a clinical trial. Now, the trial itself got really great results. So these patients, graft versus host disease is a condition that occurs in patients who receive a bone marrow transplant or similar procedure, usually to treat blood cancer like leukemia. And the initial treatment of this disease is with steroids. I should mention it's a really awful disease. It's caused by immune cells in the transplant that recognize the recipient's body as foreign. So it's like they've got somebody else's immune system inside them. It's an awful disease, and unfortunately, it leads most of these patients to pass away. The initial treatment is in steroid or is with steroids. If they fail to respond to steroids, it's called steroid resistant GVHD, and that's the population that we enrolled in our trial. We saw that 13 out of 15 showed an improvement of at least 1 grade. 8 out of 15 showed a complete response. And importantly, we saw a 2-year survival rate of 60%, which, as I'll explain in a moment, is very high in the context of this population. And importantly, we also found that the treatment was safe. So we didn't have any safety concerns or serious adverse events related to the MSC treatment. Now, just to explain a bit of background, as I mentioned already, GVHD is typically treated with steroids initially. Various other drugs, including immunosuppressants, have been investigated for treating it. Most haven't worked that well, and/or they have had safety problems. In 2019, there's a drug called ruxolitinib approved for the treatment of steroid-resistant GVHD. It got relatively good response rates in their clinical trial, but there was no apparent improvement in overall survival. And this drug is associated with a high rate of potentially serious adverse reactions. So there's still a huge unmet need here. And just to expand on that point, to compare the results we've seen to what has been shown with other therapies. So on this slide, BAT means best available therapy, which was a control group in the trial, the phase 3 trial of ruxolitinib. Rux means ruxolitinib. And what you can see is at day 28 and even more so at day 56 to 60, the response rate with our product was higher than either of the other two groups. Now, I should clarify, this is from 2 different clinical trials, so this is just for illustrative purposes, but it really is quite striking that the difference is there. And when it comes to overall survival, we saw a 60% survival rate at 18 months compared to just 36 to 38% for best available therapy of ruxolitinib. And it's worth noting that we still had a 60% survival rate at 24 months, but the 24-month survival for the other treatments was not evaluable. So it certainly couldn't have been higher, obviously, than what it was at 18 months. It may well have been lower. So these results are very, very encouraging, and we think that really shows that we have a potentially very useful treatment here. And just on the safety part of it, as I said, we haven't identified any safety issues with our MSC product. And that's a statement that actually holds true for MSCs in general, including those used, derived from bone marrow or fat and so on, used by other groups. They generally seem to be very safe. But ruxolitinib is associated with lots of really quite nasty side effects. So this table shows the grade 3 to 4 adverse reactions in their GVHD trials. Grade 3 to 4 means serious or life-threatening. And as you can see, there's quite a long list of these adverse reactions, including infections of various types, bleeding, blood clots, blood disorders. And some of these have occurred in really a very substantial proportion of patients. So that's a big problem. And again, we believe that we have a very clean safety profile. So again, that just shows that we have the opportunity to be the preferred treatment in this condition if we can continue to show the sort of results that we've seen so far in our trials. Now, moving on to diabetic foot ulcer. So in this case, we've actually got a novel MSC-seeded wound dressing, which is based on technology that we licensed from another Aussie company called TechSite Limited. And this is applied just like a standard wound dressing. In our trial, we saw in the first 16 patients in this trial, after 10 weeks, we saw a mean reduction in wound surface area of almost 88% in the active group compared to 51% in the control group. Obviously, that's very encouraging. This trial is now fully recruited. Last patient visit will be in September this year, and we're hoping to have the full and final results either late this year or early next year. So in terms of our pipeline now, as I mentioned, we have these 3 trials that are underway with results reading out between late this year and early 2026, and we're about to start the kidney transplant trial. And I'll quickly give you a summary of these trials. So the phase 2 GVHD trial is a global trial, US, Europe, and Australia. We have all of the regulatory and ethics approvals in place, including a US IND. We enrolled the first patient in March this year, and we're aiming to complete enrollment by the end of this calendar year. And if we do that, we should have the results of the primary evaluation in the second half of the next calendar year. The DFU trial I've already told you about, so we're expecting the full and final results in this one late this year or early next year. We also have a trial in osteoarthritis, which is a phase 3 trial in 320 patients. Again, this is fully recruited. This is in partnership with the University of Sydney, and it's funded by the Aussie government under an NH&MRC project grant. This trial is looking to show a disease-modifying effect in osteoarthritis of the knee. Now, there are currently no disease-modifying treatments for OA that are approved. And what that means is the treatments at the moment just treat the symptoms, right? But what we're trying to show is that we actually slow down or stop the degeneration of the cartilage in the knee. So if we can do this, this would be an absolute game changer. We're expecting the final patient visit in November next year with results a few months thereafter. So that's really going to be a huge one for both us as a company and for the wider field. The kidney transplant trial, what we're trying to do here is essentially, as most people would be aware, when you get an organ transplant, the people who receive these transplants then have to take drugs, usually for the rest of their life, to stop the organ being rejected. And in the case of kidney transplants, they typically take a type of drug called calcineurin inhibitors, which are immunosuppressants. They work very well in that they stop rejection, but they're very toxic. They cause very high rates of infection. They significantly increase the risk of cancer. And ironically, they also damage the kidneys. So there's a real move underway to try and find better ways of preventing rejection. And we believe that MSCs might do this. So the guy who's leading this trial is Professor Ton Rabelink at Leiden University Medical Center in the Netherlands. And he's previously done a similar trial using a patient's own MSCs and showed very promising results. So the aim here is that by giving MSCs, we can reduce the dose and hopefully ultimately stop using these calcineurin inhibitor drugs. And that one's just about to start really very soon. So now just to quickly finish off, we have lots of preclinical data in other conditions, most of which was conducted with very high profile research groups around the world. Lots of information about that on our website if you're interested in seeing more. We have a partnership-driven business model, so we're really seeking to out-license our products to larger companies and potentially make our platform available to other companies to do other things as well. And what this means, apart from anything else, is that we do have a potential for revenue from these deals in the relatively near term. In terms of our shareholder base, we've got two substantial holders who are Bioscience Managers with just about 13% and Fidelity International with 10%. The current market cap is around about $50 million. The price has done quite well this year, which I think is really recognition of the progress that we've been making and the news flow that's coming up as well. We have a very experienced board and management team. And just to reiterate those upcoming catalysts, so we're about to start the kidney transplant trial. We should have results from the first cohort of that late this year, complete enrollment of GVHD trial late this year, DFU trial results either late this year or early next, GVHD trial results in the second half of next year, and then followed by the osteoarthritis results in early 2026. So as I said, there's a huge amount of news flow coming. And with that, I guess I'll stop there. You know, this slide obviously just summarizes what I've run through, but I think it's an opportune time then to stop there and to move on to the Q&A.
Thank you very much, Kilian. Thoroughly enjoyed the presentation, found it very informative. Now, so I've just got a couple of questions here that have been pre-submitted by participants that I'll lead off with, and then we'll move into the ones submitted via Zoom. So first off, has there been any interest in partnering regarding SciPatch?
Yes. And just to clarify, side patch is a term used for the DFU product. And absolutely, yeah. So look, the initial data we've shown is obviously very encouraging. We've had conversations with multiple potential partners around that program. Our new Chief Business Officer, Matthias, was at Bio in San Diego last month, which is the biggest biotech partnering conference in the world. He had a lot of meetings during that time. There's numerous companies have shown interest. We've signed confidentiality agreements with some of them, and so we've got ongoing discussions there, and we're optimistic that will all lead somewhere, especially with further positive data later this year.
Okay, thank you for that update. A little bit of an interesting one here. Would it be possible to share where AI might have some significant benefits to the process and trial time reduction?
Well, that is a good question, very topical, obviously. And I would have to hold my hand up and say I'm not an expert in AI, but I have seen a number of very interesting articles and presentations about this topic. And I think it seems to be fair to say that AI, I guess, is going to have an impact on almost everything that we do currently. I don't want to say we, I mean, not just the biotech industry, but industry in general, in terms of how we work, the sort of the availability of data, the ability to process data in much shorter time frames, and potentially to conduct analyses that a simple human might take forever to do. So I think there's opportunities there. I know in somewhat slightly different spaces, there's even use of AI to help sort of design drugs and so on. That's not really relevant to us because we have a human cell, which of course is not a drug that we designed. It's rather a human cell that exists anyway, and we've got a great way of making those cells at scale. But the cells themselves are really similar to the naturally occurring ones. But I think, look, when it comes to things like just I guess improving efficiency of workflows and so on, those AI benefits are going to be there across the board. And I'm sure somebody who, unlike me, is an expert in AI might be able to talk better to how it might help improve clinical trials and so on. But as I said, that's a little bit beyond my own current knowledge.
That's all right. Thank you very much for the update. Now, moving on, is there any reason why patients treated aren't being reported as numbers 1, 2, or 3, etc., but rather as opposed to percentages?
Right. Yeah. So look, in our quarterlies, we have started reporting the ongoing recruitment numbers as percentages. And look, we just felt it was less confusing to go with a percentage because these trials are all of different sizes. So as an example, the DFU trial is 30 patients, the GVHD trial is 60, the osteoarthritis one is 320. And if we reported actual numbers, it might be quite hard to understand where we are in the process. And really the important thing is how close are we to finishing recruitment. So we felt like expressing that as a percentage was just the sort of simplest and most consistent way of doing that.
Okay, thank you for that clarification. Now that wraps up the questions pre-submitted. I'll move on to the ones that have been submitted here today. Just to start things off, are there any disadvantages of iPSCs versus embryonic stem cells?
Well, so as I mentioned, iPSCs really have very similar properties to embryonic stem cells. They actually have some advantages in some ways. For example, with an iPSC, you can get full consent from the donor. Clearly, with embryonic stem cells, that's a bit of a controversial topic. The consent typically comes from the parents and so on, but there is some controversy about that, and that is quite an important point. There have been issues in the biotech industry, for example, where cell lines that have been derived from patients there have been actions taken on behalf of those patients and so on because they didn't consent. We've got a fully consented cell line. And in terms of disadvantages of iPSCs versus embryonic stem cells, I mean, the short answer is no, not really. I mean, we as I said, they from a practical point of view, they do the same thing. They're just sort of derived in a different way. And I think overall, the reason we decided to go with iPSCs was we were able to just avoid a lot of that sort of controversy that has gone along with embryonic stem cells. And of course, people have different views on that, but the reality is there are people who feel strongly about these things, and it's not really our job to try and convince people to change their mind about what they believe. So it was just better for us to go with the iPSC version.
Well, thank you for that one. Now, moving on, has the trial design of the Part 2 AGVHD trial changed as a result of the MSB/FDA interaction?
No is the short answer. So I guess to provide a bit of context on that, that's I think a reference to Mesoblast, which is pursuing approval from the FDA for a bone marrow-derived MSC product in pediatric graft versus host disease. It's important to say our trial is an adult graft versus host disease, so they are somewhat different. A couple of reasons that is important is, first of all, adults, there are a lot more adults with GVHD than children. I think about 80 to 90% of all cases are adults. And also the drug that I mentioned, ruxolitinib, is actually not approved in children under 12. And so both of those things, the fact that there are very few children with a condition and the fact that there is no approved treatment, means the regulatory pathway might be a bit different. Now, I should stress, I have no sort of inside knowledge of that, but I'm just kind of going on what's in the public domain. But so I think they're probably in a somewhat different position in their interactions with the FDA. Our trial is a randomized controlled trial, which the FDA has made clear in public statements and in their own guidance is what they want. So we're doing the kind of trial that the FDA wants for the type of indication that we're pursuing, and really the outcome of the pediatric GVHD application doesn't really have a direct impact on that either way.
Okay, okay. Now, if the MSB gets approved for SR-AGVHD in children, how would that affect Cynata's FDA orphan drug designation as a result of a possible 7-year exclusivity period? And continuing on, would CYP001 be considered the same drug? And if so, would the approval of SR-AGVHD versus Cynata's AGVHD trial be considered the same indication?
Right. So, yeah, look, that's a very good question. And I think the key part of that question is the part about it being the same drug. And I can explain why. So basically, we have orphan designation from the FDA for our product for GVHD. And if and when we get approval from the FDA, we will get this additional 7 years exclusivity because of the orphan status. But that does not apply if somebody else gets approval for, in inverted commas, the same drug before we do. But that's where it's a key point, because our understanding is the FDA will not consider two different cellular therapies to be the same drug, in inverted commas. The reason is that cells are very complex. They can't be fully characterized. So the FDA's view is the way that you derive a cell-based product is what defines what the product itself. There's a kind of a term that the process defines the product. And so given that the FDA will not consider a different MSC product to be the same as Cynata's MSC product, it means this whole issue sort of goes away. So basically, we don't think it has any impact on us, is the short answer to that question.
Okay, okay. Thank you for that one. Now, moving on to something more stock price related. As mentioned, the CYP recent ASX releases, are we still confident that the cash runway will carry the company through to part 2 AGVHD results that will come in?
Yes. So we're we have runway that goes into the second half of next calendar year, and we are currently expecting the GVHD results to come in before that. So yeah, is the short answer.
Okay, so then moving on from that basis, once the cash runway comes in and the results come in, is there any indication of when a further on-market capital raise will need to occur?
Yeah, so look, right now we're not planning to do a cap raise right now. The reality is, in addition to having runway out into the second half of next calendar year, we also have there are options out there from last year's capital raise which expire in April. They've got a strike price of 30 cents, where they're not far off being in the money. That has the potential to bring more money in, for example. But also, as I mentioned earlier, we're actively pursuing corporate partnering deals. And so there's various ways that our cash position could be strengthened. Obviously, capital raising is one of them, but there are other ones, and we're not in a rush to do anything right now.
Okay. Okay. Now, just focusing back on a different topic. One of the participants understands that the original Wharf licensing agreement has been adjusted in regards to first product to be approved by. Can it be shared by the new what the new date is as stated in the amended contract, and which indication do you expect to have the best chance to first be approved?
Yeah, so look, I mean, I obviously can't share non-public information in this forum. We have a very strong relationship with Wharf. We do have milestone obligations under our agreement, as is typical for an agreement like this. We believe that we can, that we can fully manage that situation, and we're not expecting that to become a problem for us. In terms of the indication that's likely to be the first approved, there's a couple of possibilities really. GVHD is clearly one of them, but also osteoarthritis potentially, depending on the exact timing of the results and the strength of the results. There's a few different factors at play there, but yeah, I suppose as we sit here right now, the two leading ones would be GVHD and osteoarthritis.
Okay. Okay. Now, relating to the survival rate histograms you showed throughout the presentation, when comparing the overall survival throughout the the three ones in particular, Is that based on matched patient groups, or is there potential comparing of grades CD with grades B and D?
So as I mentioned while I was going through the slides, and I think it's clarified in the footnote, those trials were different trials. So we didn't have a head-to-head trial with ruxolitinib or the other agents. So it's really there for illustrative purposes. The inclusion criteria were broadly similar, but of course the sample size, again, as clarified in the footnote, is different. So the patients aren't matched, no. But it's really just to give an illustrative idea of the fact that a 60% overall survival rate at 2 years is exceptionally high in the context of these patients, right? And of course, we will see as our trials move forward with our larger trials, hopefully we'll see similar outcomes. But we're certainly not trying to portray those as matched comparisons at this point.
Of course, of course. Now, what stage of development are your competitors in, and what edge does CYP potentially hold over your current competitors?
So, look, I suppose it depends what sort of perspective somebody has about a competitor. I mean, if we look at each individual clinical indication, of course, we have on the one hand, we've got other MSC type products, but we've also got other products that might not be cell-based products. That could be anything that are being developed for the same indication. So, of course, that's there's lots of drugs in development for any indication at any given time. But I think really the way we see it is when it comes to MSC based products, we believe that the manufacturing technology that we have gives us really the best chance of being able to make consistent and high quality MSCs at large scale.
Okay.
And when it comes to, if you like, a potential competitor that's not an MSC or not a cell therapy, well, as I mentioned during the presentation there, one important point is that MSCs appear to be extremely safe. There's really no known safety concerns with them. And that's almost unheard of when you go into the sort of the conventional drug or monoclonal antibody space. So most other products do have known adverse reactions associated with them. And so that in itself is a big sort of general advantage that we would have over competitors. But as I said, you've really got to get down to it in terms of each indication and each, the landscape for each of them. And we're certainly doing a lot of work to look at that at the moment.
Of course. Now, how far is the technology transfer from FCDI? It has been mentioned years ago that it has started, but now in 2024, we're still being fed. I was still being told the same, despite in 2017, the Streetwise interview with Stuart Washer, that it was quite a simple process.
Look, I don't know what the reference to 2017 is. I mean, we signed the strategic manufacturing partnership agreement with Fujifilm in December 2021. So the tech transfer process got underway during 2022. And we've so far we have made a lot of progress there. We have made demonstration batches of the product and so on. So, we're certainly pleased with the relationship we have with Fujifilm. They've got a really very deep expertise in iPSC-based therapies. CDI, the company that we're directly interacting with, which is now a wholly owned subsidiary of Fujifilm, CDI was actually founded as an iPSC company, initially making iPSC-derived cells for research purposes before they started moving into therapeutic cells like ours. But what that means is there's probably no other company in the world that has the same sort of level of experience and expertise when it comes to iPSC. So we're very happy with all of that.
Okay. Now, with the commercial partnering business model that has been in place since day one, the only licensee Fujifilm has had a back the license. What is, in your eyes, the main reason for Cynata not being able to get another licensee?
Right. So look, the original Fujifilm license deal that we did, it's important to say, was where we out-licensed our technology to Fujifilm. So they were going to become the licensee of Cynata, who was responsible for developing and ultimately marketing our GVHD product. And Fujifilm really had a pretty widely publicized change in strategy since then. They're no longer, as a rule, pursuing their own therapeutic products, but rather they're positioning themselves as a manufacturing player. So they've acquired businesses really at all stages of that manufacturing sort of piece. They've acquired reagent and media companies, contract manufacturers, etc. And so that was really the background to them renegotiating, or us renegotiating, the agreement with them, under which, as I said, in late 2021, they returned the rights to GVHD to us and became our strategic manufacturing partner. I think why have we not done another deal since? Well, we have certainly, as I mentioned earlier, we've got ongoing conversations, but we've also got an ongoing phase 2 clinical trial right now, right? So there's sort of a process to go through with the partners is one part of this. But the other thing is we've also got one eye on the sort of the value of a deal and the additional value that we can create by adding phase 2 results to the data package that we already have. So we are very optimistic that we will do another deal in GVHD, but we want it to be the right deal.
Oh, well, that's good to hear. Now, has the Simris process been tested with ISPC from a different provider that's not CDI or FCDI?
Not in a sort of a good manufacturing practice setting, in which, we haven't made clinical product using iPSCs from another provider, but certainly in the early days when we were doing development work, we did indeed use iPSCs from other sources. So we're quite confident that it would work with iPSC lines from elsewhere if that was relevant. Yeah.
Okay. Well, we've got a participant that asked, congratulations for the efficacy result on CYP 001. Will it have any cost advantage when compared to ruxolitinib treatment?
So in terms of cost advantage, look, that's a slightly difficult question to answer right now because we haven't sort of put a price on our product as yet. Ruxolitinib, it should be noted, is not a cheap treatment either. It is a relatively expensive product, and unlike our product, it needs to be taken on an ongoing basis. So sometimes people look at the cost of ruxolitinib is tablets, right? Some people look at the cost of one box of tablets and it's about, I believe something like $6,000. But the reality is you've got to take it on an ongoing basis. So the cost of that actually adds up. Look, I wouldn't sit here today and say that I expect us to have a significant cost advantage over ruxolitinib, but I'm not sure if the reverse will be true either, if that makes sense. But I think where our advantages really lie is hopefully better efficacy, but I'm almost certain that we'll have a better safety profile, and they're very important.
Of course. Well, that nearly wraps things up. I've got one last one here. Could we get insight into what the incentives are for management compensation in the upcoming financial year?
Well, we are currently going through that process right now. So I can't answer that question because it hasn't actually been decided yet. But look, needless to say, there are KPIs set by the board and for myself, obviously, and then that flows down to the rest of the team. And those KPIs, they are centered around, I guess, what most people would expect them to be centered around, which includes things like, hitting milestones in our clinical trial programs, commercial partnering, and finance and share price related metrics. So I think, they would be what people would be expecting to see.
Okay. Well, thank you very much for that clarification. We've just had one more that has come through. Regarding the milestones you've just mentioned, will these milestones specifically be mentioned to the public, or will they remain a contract basis to the relevant people?
So we're not planning to announce all of the individual KPIs, but what we are planning to do is, certainly in the annual report, is to give a high level summary of what the KPIs are and so on. Yeah.
Okay. Well, that about wraps everything up. Kilian, is there anything you'd like to add?
No, look, just thanks again for the opportunity to talk, and thanks to everybody for joining. And especially there was lots of questions there, which is great to see. It's always reassuring when we've got an engaged audience like that. So I appreciate that, and we look forward to delivering a lot more good news in the months to come.
It is. Thank you very much, ladies and gentlemen, for attending, and thank you very much, Dr. Kilian, for your time. If anyone would like to review this presentation webinar or distribute it to anyone in particular, it'll be uploaded on the Sharewise YouTube within a couple of hours. So thank you very much, Kilian, and thank you very much, ladies and gentlemen. Enjoy the rest of your day. Cheers.
Thank you.